Hello, my name is Jonathan Lamarkin. I am one of your Bio 5A SIs for the quarter. If you ever need any help, please attend one of my sessions or any of the SIs. So we're going to go straight into it. Lecture 8, Cell Membranes, Part 2. So we know that there are two specific types of cells that we explained in class. There are prokaryotes and there are eukaryotes. An example of a prokaryote can be bacteria. An example of eukaryotes can be animal or plant cells, and each one has a cell membrane, also known as a plasma membrane. As shown here, they both have plasma membranes. Okay? And each cell membrane or plasma membrane consists of three things, which is the phospholipid bilayer. We went over this in the lipid lecture, where it contains hydrophilic heads and hydrophobic tails, embedded proteins, and cytoskeleton attachments. The embedded proteins allow specific molecules to pass through the phospholipid bilayer, and the cytoskeleton attachments help form its shape and its structure. As shown here, there are some examples, and we're going to work as a class together to see if specific molecules can pass through. So oxygen, O2, is it polar or nonpolar? First, we have to figure out the phospholipid bilayer. It has hydrophilic heads, and the hydrophilic heads, we know hydrophilic means polar, and we know hydrophobic means nonpolar. So if we had an O2 molecule, would it be able to pass through? And the answer is yes, because it is nonpolar and it is also small. And we know small and nonpolar molecules can pass through. CO2, is it nonpolar or small? It is nonpolar and it is small, so it will be able to pass through. C, H2O, is it small or big? Well, it is small, so it can technically pass through, but it will pass through very slowly. Therefore, it would need an embedded protein called aquaporins, allowing it to pass through. So yes, it can pass through. However, it's very slow, so it needs aquaporins. So this would kind of be like half a check mark. So realistically, we would want aquaporins. So in reality, it would be an X. It would need aquaporins to transfer a lot quicker. The next thing would be glucose, C6H12O6. Will it be able to pass through? No, because it's a polar molecule and it's very big. Therefore, it would need something called a carrier protein. For example, pretend we had a carrier protein like this, and then glucose itself wouldn't be able to go through, but with the carrier protein, it will allow it to pass through. The next thing is NaCl. NaCl, we know, splits into Na plus and Cl minus, which are two ions, and ions are polar. Therefore, it is attracted to the hydrophilic head. So it by itself, it would not be able to pass through, but it would need something called a channel protein to pass through. So just like glucose, pretend this was a channel protein, and then Na plus and Cl minus, they would need help with a channel protein. So channel proteins refer to ions, and then carrier proteins refer to bigger molecules that are polar. The next thing we went over is passive versus active transport. So as review, passive transport is movement of substances across cell membranes without need of energy input. There are actually three different examples of passive transport. The first one is simple diffusion or diffusion. That's when molecules diffuse down concentration gradients. So for example, there has to be a semi-permeable membrane. So for example, on the side on the left, we have 6 molecules and we have 2 molecules on the right. And diffusion would want to make something called equilibrium, and it's going to go down the concentrated gradient. So the molecules are going to go on the right side until there's equilibrium, and as shown on the right side, there is equilibrium. The molecules itself will continuously move back and forth, but there will always be equilibrium. And this requires no energy. The second type we learned is facilitated diffusion, and it is also diffusion, but as the word facilitated means the help of doing something. In facilitated diffusion, it uses 2 different proteins as we discussed before, channel proteins and carrier proteins. Channel proteins are specific to ions, and carrier proteins allow large molecules to pass through. For example, again, carrier would be something like glucose, and then channel would be specific ions, such as Na+, Cl-, etc. The next thing we went over was osmosis, and instead of the solutes moving back and forth, we're actually looking at the solvent. So osmosis occurs when the solute isn't able to move, but the solvent itself, which most of the time is water, it'll be able to move back and forth. As shown here, there's a concentration of 4 molecules on the left side and there are 8 on the right side. Normally, if it was just diffusion, the molecules on the left would move to the right. However, since the molecules cannot move, then osmosis will occur. And this only occurs if there's aquaporins. Aquaporins help water move a lot quicker. Therefore, the water would move to the right, as shown here, and the concentrations would be very similar. And we learned in Chem 1A that concentration equals molarity, and molarity equals moles over liter. So you guys don't really need to know the specifics for this class, but just know that we want to have the same molarity on both sides. Okay? Here are some examples of osmosis, but we need to understand what these definitions are. Hypertonic, as stated before, means more concentrated and more solute present. Hypotonic means less concentrated, less solute present, and isotonic normally means equilibrium. So hypertonic itself, if we look at the top, hypertonic solution, that means it's referring to the solution itself. Hypertonic means more concentrated. So on the outside we could see it's very concentrated, but on the inside it's not concentrated or less concentrated. Therefore the water or the solvent in the inside is going to want to move out. For the second example, hypotonic solution, on the outside it's not concentrated. Not concentrated. On the inside it is concentrated. Therefore the water is going to want to move to the inside. And in doing so, it's going to expand the cell. And the last one is isotonic. And isotonic is pretty much equilibrium. The concentration on the outside and the inside are equivalently the same. Therefore, they're going to move back and forth. Okay? The next thing we went over is active transport. And active transport moves up or against the gradient. That's extremely important. Moves up or against. That's extremely important. Against the gradient. And it requires energy. For this class, we mostly use ATP as the energy source. So why do we do this? So for example, if we looked at this example here, the side on the left has 2 molecules and the side on the right has 4 molecules. However, if we knew diffusion, we would think that the molecules would move to the left. However, we're not looking at diffusion. We're looking at active transport, so the molecules are going to want to move to the right. And the only way it could do that is if energy is present. If energy is not present, then this wouldn't occur, and diffusion would just occur. So as shown here, the one molecule is going to go to the right side, therefore the concentration gradient, it will move against the concentration gradient. So now there's 1 and 5. But why? Why would we want to do that? That doesn't make any sense. Why? The reason why is to make an electrochemical gradient, and therefore you're going to increase the potential energy. And as we learn, or you guys will learn in the future, if you increase the potential energy, then you're able to use kinetic energy in the future. So if you do this, then you can couple this to do work in another reaction. So an example here is we learned in class. For example, if we inserted ATP and we moved the hydrogen ion to the right side, now we have a gradient. And therefore we're able to pair this H plus with sucrose to this co-transporter, and now they're both together are going to move to the left side. So at the top, this is active transport. But on the bottom, since we already did active transport, the cell wants to maintain homeostasis, equilibrium. So the bottom would be considered passive transport. So H plus is going to move back into the cell with the sucrose molecule. So most of the time active transport leads to passive transport, but there's a reason why. If there was no hydrogens on the right side, then the sucrose wouldn't be able to transfer to the left side. The next thing we went over were the different types of active transports, and it falls under the definition of bulk transport, which is transporting large materials across the membrane. And the first one is exocytosis, which is a simple concept to understand because exo sounds like exit, so we know it's just going to exit. So that's when a lot of molecules aren't able to pass through the cell membrane without energy or proteins, therefore it needs ATP. So for example here, there's a lot of molecules here, and it's going to enter something called a vesicle, and the vesicle is going to fuse to the phospholipid bilayer, changing its shape, and once it attaches to the phospholipid bilayer, the molecules are able to leave. And this requires ATP. That's extremely important. Without ATP, this wouldn't occur. The next one is endocytosis. So we know exocytosis means leaving. Endocytosis means to enter the cell. And there are 3 different types of endocytosis. The first one is phagocytosis. And phagocytosis is pretty much the cell eating something. As shown on the left side, it's going to eat a big particle and it's going to enter into a vacuole. And this will be considered a food vacuole. And this food vacuole will then meet up with the lysosome, and the lysosome is going to digest it. And in the next lecture, we'll actually go more into detail what lysosomes are and the different organelles. The second one is pinocytosis. The way I remember it, P-I-N-O sounds like pint. And we know pint is referring to like 2 cups of water. And for some reason, I just think pinocytosis is a pint. And it's just like cell drinking. So for the second example, it's pretty much looking at different fluids and it's taking it in. So the first example, you're eating solid. The second example is pretty much fluids. And the third example is receptor-mediated endocytosis. So you're going to have receptors that are specific, as shown here. There are some receptors here, and they're specific only to the stars. Therefore, once the molecules bond to these receptors, they're able to do something similar to phagocytosis, where they enter this vesicle and it goes inside the cell. So the last thing we went over was cell structure, and I'm not going to go too into detail, but I'm going to do a little summary. So we know all cell structures, they have a cell membrane, a cytoplasm, and region with DNA and RNA. So if we look at the left side, it's a eukaryote cell, and the right side, prokaryotic cell. So they both have cell membranes, or also known as plasma membranes. They have a cytoplasm. So cytoplasm is the region between the nucleus and the cell membranes. So for the left side, it could pretty much be this all the way to this, and it includes the organelles. But if we want to go even more into detail, we would look into something called a cytosol. And the cytosol is pretty much just the liquid in between the nucleus and the cell membrane, but then the cytoplasm is everything included. And then the third thing is region with DNA. We know eukaryotic cells, the DNA is in something called a nucleus, which is very compacted. However, in a prokaryotic cell, the DNA is in a region called nucleoid, and it's not in a nucleus, so the DNA kind of roams around by itself. So why is DNA important in eukaryotic cells? DNA is made into RNA and made into proteins. DNA in eukaryotic cells are found in the nucleus, and DNA itself can't leave the nucleus. Therefore, DNA will be converted into RNA, or even more specific, something called mRNA, which is in the nucleus. And the mRNA is going to go outside the cell to meet something called ribosomes. And ribosomes can be found in 2 areas. It could be found by itself in the cytosol or the cytoplasm, or it could be found on something called the rough endoplasmic reticulum. And we'll go more over this in class. But ribosomes pretty much translate the mRNA into amino acids, which become polypeptide chains, as we learned before. And these polypeptide chains become protein. And proteins are essential for many things in our day-to-day lives inside our body. So if it's by itself, that means the protein is going to actually stay inside the cell. Stay in the cell. And if the mRNA goes into ribosomes in the rough ER is going to transfer it to the Golgi apparatus, and the Golgi apparatus is going to transfer the protein outside the cell. Okay. And yeah, if you have any questions, please come to my SI or come to any of the other SIs, and we'll gladly help you out. Thank you so much, and I hope you have a great day.